Cell injury. Cell is the fundamental unit of life. A group of cells form a tissue. A collection of tissues form an organ. Many organs form an organ system. And many organ systems form an organism. Cell at its basic level is divided into two types: epithelial cells and mesenchymal cells. Most forms of disease starts with cell injury, which is followed by gradual or rapid loss of its specific function. Cell injury occurs when cells are subjected to severe stress that they are no longer able to adapt, or when the cells are exposed to inherently damaging agents or suffer from intrinsic abnormalities. The cellular response to stress may vary and depends upon two variables: host factors, the type of cell and tissue involved, nature and extent of injurious agents. Cellular response to cell injury may comprise the following. When there is an alteration in functional demand, the cells may adapt to the changes which are morphologically expressed. These changes revert back to normal after the stress is removed. When the stress is mild to moderate, the injured cell may recover after the stress is removed, while severe forms of cell injury may lead to cell death. After the removal of stress that has caused the reversible cell injury, the residual effect may persist as evidence of cell injury at subcellular level. Subcellular changes or metabolite may accumulate within the cell, that is, intracellular accumulations. Disease processes at cellular level may be understood by studying the causes and mechanisms of cell injury and various cellular adaptations in response to stress. Cell injury may be caused by either genetic or acquired causes. Genetic causes may be further classified into developmental defects, cytogenetic abnormalities, single gene defects, multifactorial inheritance disorders, storage diseases, or inborn errors of metabolism. Developmental defects. They are a group of abnormalities that occur due to errors in morphogenesis during fetal life. Example: anencephaly and spina bifida complex. Cytogenetic abnormalities. Abnormalities in chromosomes can be divided into two types: numerical abnormalities, example, polyploidy and aneuploidy. Structural abnormality, example, translocations, deletions, inversion, ring chromosome, and isochromosome. Single gene defects. These disorders are due to mutation of a single gene. There are four types: point mutation, nonsense mutation, frameshift mutation, and trinucleotide repeat mutation. Multifactorial inheritance. They are a group of disorders caused due to the combined effect of genetic composition and environmental influences. Example: cleft lip and cleft palate, diabetes mellitus, and hypertension. Storage diseases and inborn errors of metabolism. They are a group of disorders that occur due to genetic defect in the metabolism of carbohydrates, proteins, and lipids. resulting in intracellular accumulation of metabolites. Example: glycogen storage diseases and mucopolysaccharidosis. Acquired causes may be further classified based on the underlying agent: hypoxia and ischemia. Physical agents, chemical agents, microbial agent, immunologic agents, nutritional agent. Aging, psychogenic diseases, iatrogenic diseases, idiopathic diseases. Hypoxia and ischemia. Cells require oxygen to carry out various biochemical reactions that generate energy and performs various metabolic functions. Deficiency of oxygen or hypoxia will result in the inability of cells to carry out its basic activities. Hypoxia is the most common cause of cell injury. It may result from reduced supply of blood due to interruption or ischemia. It may also result due to disorders in oxygen-carrying RBC. Example: anemia, carbon monoxide poisoning, heart disease, lung disease, and increased tissue demand. Physical agents. Physical agents that can bring about cell injury are as follows: mechanical trauma, thermal trauma, electricity, radiation, rapid changes in temperature. Chemicals and drugs. Chemicals and drugs. The various chemical agents and drugs that can cause cell injury are chemical poisons such as cyanide, arsenic, mercury, strong acids and alkalis, environmental pollutions, pesticides and insecticides, oxygen at high concentration, hypertonic glucose and salt solutions, social agents such as alcohol and narcotic drugs, therapeutic administration of drugs. Microbial agents. Infections caused by bacteria, viruses, fungi, protozoa, metazoa, and other parasites that result in cell injury. Immunologic agents. Immunity protects the organism against various injurious agents like pathogens. This system may lose its regulatory control under some conditions, causing cell injury to the very organism that it is supposed to protect. Example: hypersensitivity reaction, anaphylactic reactions, autoimmune diseases, etc. Nutritional derangements. Nutritional derangements. A relative lack or excess of nutrition can cause nutritional imbalances. This may include nutritional deficiency diseases due to overall deficiency of nutrients. Example: starvation, or protein-calorie. Example: marasmus and kwashiorkor, or minerals or trace elements. Example: anemia. Nutritional excess seen in affluent societies resulting in obesity, atherosclerosis, heart disease and hypertension. Aging. Cellular aging due to senility ultimately leads to loss of ability of the cell to perform normal function, undergo replication and repair. This leads to cell death and ultimately resulting in the death of the individual. Psychogenic diseases. Commonly acquired mental diseases like depression, schizophrenia, caused due to mental stress, strain, anxiety, overwork and frustration does not manifest with specific biochemical or morphological changes. Iatrogenic diseases. These are diseases or deaths caused due to error in judgment by the physician and side effects caused due to wrong drugs. Example: iatrogenic transfusion. Idiopathic diseases. These are diseases due to unknown causes. The exact cause for few diseases are still not understood. Example: idiopathic urticaria. Pathogenesis of cell injury. Injury to a normal cell may result in a state of reversible or irreversible cell injury which have varied biochemical alterations. The following scheme applies to most forms of cell injury by various agents. Factors pertaining to etiologic agent and host. The outcome of cell injury depends on the host cells and the etiologic agent concerned. Type, duration, and severity of injurious agent. The extent of cell injury depends upon the type, duration, and severity of the stress. Example: small dose of chemical toxin or ischemia for a short while can cause reversible cell injury, whereas large dose of toxin or persistent ischemia may cause cell death. Type, state, and adaptability of target cell. The type of cell involved in the stress, its susceptibility to stress injury, its nutritional and metabolic status, and its ability to adapt to hostile environment determine the extent of cell injury. Example: the oxygen dependency of skeletal and cardiac muscle. Skeletal muscle can withstand extended periods of hypoxia. Whereas cardiac muscle suffers irreversible cell injury. Due to coronary occlusion within 20 minutes. Common underlying mechanism. The following essential intracellular biochemical phenomena underlie all forms of cell injury. ATP depletion due to mitochondrial damage. Deranged metabolism and faulty transmembrane exchanges due to damage to cell membrane. Usual morphologic changes. One of the first signs of cell injury is the change in the biochemical and molecular aspect of the cell. These are associated with ultrastructural changes in the injured cells. Eventually, changes in morphology of cells and organ appear in gross and light microscopy. The morphologic changes of reversible injury, that is, hydropic swelling, appear earlier, while that of irreversible injury or cell death appear later. Example: myocardial infarction. Functional implication and disease outcome. Any cell injury, whether reversible or irreversible, adversely affects the cell resulting in derangement of its function. As a result of this, clinical features in the form of signs and symptoms would appear. The outcome of the disease will depend upon the response of the disease to various treatment processes as against the biologic behavior of the disease. Reversible cell injury. The most common form of cell injury is hypoxia and ischemia. If they are of a short duration, the effects may be reversible on immediate restoration of circulation. The sequence of biochemical and ultrastructural changes in reversible cell injury are as follows. Decreased generation of cellular ATP. ATP generation by aerobic or anaerobic respiration are of two ways in which cells meet their energy demands. Ischemia due to interruption in blood supply as well as hypoxia due to other causes reduces the supply of oxygen to the cells, thereby decreasing the net production of ATP. Ischemia due to interruption of blood supply results not only in decreased oxygen supply but also a reduction in glucose availability. This results in a more severe and rapid form of cell injury apart from accumulation of metabolic waste products. Hypoxia from other causes result only in decreased oxygen supply, whereas glucose supply is unaffected, thereby allowing the cell to switch over to anaerobic glycolysis. The cell injury is less severe. However, myocardium, proximal tubular cells of kidney and neurons of CNS are solely dependent on aerobic respiration, and thus cell injury due to ischemia are more severe and rapid in these tissues. Intracellular lactic acidosis, nuclear clumping. Due to the switch from aerobic to anaerobic respiration as a result of ischemia or hypoxia, there is a depletion of glycogen and accumulation of lactic acid, lowering the intracellular pH. Initial fall in intracellular pH causes clumping of nuclear chromatin. Plasma membrane pump damage, hydropic swelling. Continuous generation of phospholipids from cellular fatty acids require ATP and are essential to maintain the integrity of cell membrane. Lack of ATP interferes with this process, resulting in damage to membrane pumps involved in regulation of sodium, potassium, and calcium levels. The accumulation of sodium inside the cell leads to an increase in intracellular water, resulting in hydropic swelling of the cell. Failure of calcium pump. Decreased cellular ATP lowers the activity of calcium pump, resulting in increased influx of calcium into the cells, particularly the mitochondria, causing its swelling and deposition of phospholipid-rich amorphous densities. Reduced protein synthesis. Chronic hypoxia causes the membranes of endoplasmic reticulum and Golgi apparatus to swell up. Ribosomes are detached from rough endoplasmic reticulum. The net effect of this is reduced protein synthesis in cytoplasm and Golgi bodies. Removal of acute stress or assault can restore the cell to normal state.